Ready to score 360 out of 360 in NEET Bio within 250 days? I welcome you all to the series, and today we will be talking about the second last part of this particular chapter, which includes chromosomal and Mendelian disorders. Children, this is the most important topic of this chapter, so we cannot afford to miss this for sure. Keep your ears and eyes, everything open, because we definitely have to put a lot of attention, and it's a very interesting topic as well. You will get to know about lots and lots of diseases. Yeah, so what did we cover in the previous session? We spoke about pleiotropy, we spoke about polygeny, we spoke about sex determination, all of these things with examples, with the diseases associated with what all things are there, extra, and what all type of questions could also be framed. Now, if you want the notes for the same, you can join our WhatsApp channel. It just includes us, the NEET people. Yeah. Now, going ahead, children, the number of questions that came from this chapter was 6 in the previous year. So, 6 into 4, 24. Okay, 24 questions from this chapter. So, that's why I said it's a high weightage chapter. I cannot just say it's a high weightage chapter. This chapter holds the most amount of marks in NEET usually. So, previous to previous year, it was 8 questions. Now 8 into 4 you can calculate. That's many mark you could lose if you did not study this particular topic. Now going ahead, I gave you homework if I remember, but if you remember, do let me know the answer as well. If you have not yet told the answers, you still have time. If you're watching this, tell me the answer. Now, if you're not able to understand the question, let me try. Which of the following pairs is wrongly matched? You have to find the wrong matches. Okay, so starch synthesis in P says multiple alleles are there for starch synthesis in P's. Next thing, ABO blood grouping shows codominance. Okay, XO type sex determination is found in grasshopper. T.H. Morgan was the one who found out linkage. So according to me, children, everything is correct. ABO blood group shows codominance. You know that A and B, both of them, they become dominant together, which is why we have AB blood group as well. Now, XO type of sex determination is found in grasshopper, because grasshopper is an insect and most insects have this type of sex determination. Okay, where they get X from one and they get nothing, no chromosome from another gamete, and when they fuse, you get a male. So XO, whoever has this condition, that will be a male grasshopper or male insect. Okay, now going ahead, T.H. Morgan was the one who found out linkage as well. Talking about starch synthesis in P, it is given as an example of multiple alleles. No, it is not the example of multiple alleles, children. Okay, first thing, it is an example of pleiotropy. This is an example of the alleles which controls it. It's an example of pleiotropy. That is one very important thing. Apart from that, there is also one more thing. Apart from that, they do not show multiple alleles. There is only one allele or one pair of allele that will be coordinating this. That is capital B, and then you have small b. There is no other things that will be supporting or helping with starch synthesis. So you have to understand that. Okay, there are only 2 alleles. That is a pair of alleles. That is the only thing. So this is wrong. So this is not the example of multiple alleles. And also one more thing, they show incomplete dominance. So capital B is incompletely dominant over small b, which is why the one that have capital B, small b genotype will be having intermediate starch synthesis. Okay, so if it is capital B, they show high amount of starch synthesis. If it is small b, it shows low amount of starch synthesis, and when it is capital B, small b, it shows intermediate amount of starch synthesis. Now just a second, because the pen I think has some problem. Just give me a second. Okay, I changed the pen. Now going ahead with today's topic that we have, let's see. Mendelian disorders. What do you mean by Mendelian disorders? Now, whenever there is a problem within the genes, okay, when there is a problem within the genes, children, you call this as Mendelian disorders. Now, I have one more category of disorders which you call as chromosomal disorders. Wherefore, we have example is that if I was supposed to have 1 chromosome, instead of that, I have 2 chromosomes. Okay. I am adding the chromosome, or there can be a case where I should be having 1 chromosome, but I do not even have that 1 chromosome as well. That is also going to lead to some other disorder. So anything that deals with the number of chromosomes and stuff like that, you call them chromosomal disorders. But when you're talking about some problems within the genes that are present on the chromosome. For example, this gene that is there, it is mutated, it has some problem in it, it is not doing its normal function. Then, and the disease which is caused by this problem is known as a Mendelian disorder. Okay, it will be known as a Mendelian disorder. So I hope you got it. Whenever you are talking about the number of chromosomes, that it is less or it is more, you say that it is caused due to a chromosomal disorder. But when you say that there is a problem with the gene that is present on the chromosome, it will be what? Categorized under Mendelian disorders. Fine. Now, let's begin. So you have two types, one is sex linked and the other one is autosomal. So I hope, children, I hope that you know this, that in our body, for example, we say that we have 23 pair of chromosomes, right? We say that we have 23 pair of chromosomes. So in that, 22 are the one which will be responsible for all our physical characters and stuff like that. Well, when we talk about the 23rd pair, it is mostly responsible for the physical characters as well as the sex. It is also responsible for our sex. So I say in my body, I have two chromosomes. This is the 23rd pair of chromosomes. There has to be a pair. Okay. So I will have an X chromosome and the other one will also be X, which is why I am a female. Okay. Suppose there was a person who had one X chromosome and the other one was Y. Then this person has to be a male. Now, what do you mean by sex-linked Mendelian disorders? When there are problems on the genes which are present on the sex chromosomes. Okay, the ones where there are problems present on the genes of these sex chromosomes, you call it sex-linked Mendelian disorders. Okay, and the sex chromosome, you all of them together, what do you call them, children? You call them, where shall I write? Okay, just give me a second. I'll remove this. Okay, and I'll write it here. So, the 22 pair of chromosomes, okay, the 22 pair of chromosomes which are there for our normal physical characters but does not define our sex, they are known as autosomes. They are known as autosomes. Whereas the 23rd pair, which is responsible for determining our sex, this is known as allosome. Fine. So these are autosome and these are allosomes. Now, sex links means we are talking about the 23rd pair. We are talking about that there are certain defects on the 23rd pair of chromosome, which is why we are having this particular disease. Now again, if there are defects on the other chromosome, that is the other 22 pairs of chromosome, any defect is there, you call it autosomal disorder. Okay, you call it autosomal disorder. Now going ahead, you can subcategorize them into dominant and the other one is recessive. Okay, similarly over there also. Okay, so examples are also given over here, that is, for recessive, I say sex-linked recessive disorder is color blindness and hemophilia. But ma'am, I don't understand what's dominant and what's recessive. So children, please listen to me very carefully, okay? Suppose I have chromosomes over here and I say that color blindness is a sex-linked recessive disorder. That means probably if I have X and I have X, obviously, okay, it's there on X chromosome. The problem is there in the X chromosome, not on the Y chromosome, okay. Both of them, they are X, they are also present on, problem is in the X chromosome, not the Y chromosome. Now, so XX is there, cool. I am saying the gene over here, it is getting mutated. So this is the mutated gene, but I have a normal one. So this is normal one and this is the mutated one. I am writing C over here because it is for color blindness. Now, will this female who has X and X chromosome have this particular disease, that is color blindness? No, because this is a sex-linked recessive disease. That means this guy, if it is present like this, just one number is there, then the other one which is normal will be dominant over this, and hence the person who is having this particular disease is just a carrier but do not show the disease. So that female would not be showing off this particular disease. On the other hand, if I say the condition was like this, where there is chromosome X and X, okay, this X and X chromosome, and the gene is present, the problematic gene is present both the times, okay, in both the chromosomes you have the problematic gene, then obviously there is no one to dominate over it, hence that person will be colorblind. Okay, But ma'am, if this is sex-linked recessive disease, then what do you mean by sex-linked dominant disease? So you know, same condition I am putting over here. So there is a chromosome, there is a chromosome. Okay. Now I am saying that there is a normal gene, but there is one chromosome that is having this particular problem, whatever problem it is. Okay, there is no example for dominant sex-linked dominant diseases in your book. Okay, so now just suppose that this gene is affected and this disease is a sex-linked dominant disease. So basically I just need one copy, and then also what will happen, the normal won't be able to overshadow me. I will show that effect, and this particular person who is there will be having that particular disease. It won't be the carrier, he or she would be having that disease, because this one particular gene is enough to show the character. This is what you mean by sex-linked dominant. So over here it was not like that. One was not enough because the other one was normal and other one was dominant over this. So this is a recessive one. Recessive ones show their character when both of them are same. That's when they show their character. So hemophilia as well as color blindness basically are sex-linked recessive diseases, children. On the other hand, if we talk about other disease that is present over here, for autosomal, again they have dominant. Okay, example is cystic fibrosis, which is not given in your book. Okay, then over here for recessive you have two things that is given in your book. Let's see, that is sickle cell anemia, sorry, three things that is given in your book and one that is outside. So sickle cell anemia, thalassemia, phenylketonuria and muscular dystrophy, all of them, they are autosomal recessive diseases. As in, I am trying to say that if one chromosome is normal but the other chromosome is having this particular mutation, I am writing mutation over here, then this particular gene which is normal will be showing its effect, and hence that person would not be giving off this particular disease, would not be showing the disease. But on the other hand, if there is a pair of chromosomes and both of them has this mutation, in this condition definitely that person will be getting this particular disease or showing this particular disease, that is autosomal recessive diseases. So it's on autosomal chromosomes, not on the sex chromosomes. That's the only difference. Okay, it's not on the sex chromosome, but this is on the sex chromosome. Example is sickle cell anemia, thalassemia, phenylketonuria and muscular dystrophy. Muscular dystrophy and cystic fibrosis is not given in your book, but I saw the questions, I saw that they have given this as one. So for example, eliminate the one which is not autosomal recessive. So, and cystic fibrosis is given over there. So you know it's not autosomal recessive, it is what? Autosomal dominant. But rest of things that they are given. So either you know this or you know this, but you should be knowing everything. So I hope that you caught the basic concept of this. Mendelian disorder is when you have a problem with the gene that is there on the chromosome. Okay, that is what causes Mendelian disorders. Chromosomal disorders are when the number of chromosomes are somehow different from the normal numbers. Okay, so instead of having 23 pair of chromosomes, you can have 24. So one chromosome might come as an extra chromosome. So that is a problem because we don't need it. Each chromosome has genes which will be coding for proteins, and that protein will be defining us. So we don't want anything extra in our body. Okay. Now there are two types of Mendelian disorders, sex-linked and autosomal. The problem that occurs because of the mutations on the genes that are present on the sex chromosome, basically X chromosome, they are known as sex-linked diseases. Now you have dominant where even one copy if is there, muted copy is there, then also the disease would be occurring. On the other hand, if you have just one copy, then you might be a carrier, you might not show that disease. Okay, for example, it happens in color blindness and hemophilia. These are examples of sex-linked recessive ones. On the other hand, you have autosomal dominant and autosomal recessive. Dominant one is cystic fibrosis. The recessive ones are this, that is sickle cell anemia, thalassemia, phenylketonuria and muscular dystrophy. Got it? This is not sex-linked. Now, one by one, we will study about each of them. So let's start with color blindness first. Okay. Okay. There's one question over here. Depending on what we studied as of now, you can put one as the question number and you can tell me the answer if you know it already. Okay, so pick out the correct statement. Hemophilia is sex-linked recessive disease. Okay, Down syndrome is due to aneuploidy. Phenylketonuria is autosomal recessive disorder. Sickle cell anemia is X-linked recessive gene disorder. So you have to find the correct statement. So tell me the answer for the same. I think whatever we studied, it is enough for giving you the answer for this particular question. So put one and write the answer. Can you do it, my children? Okay. Now going ahead, let's talk about color blindness and then we will write everything. Okay, and study. So it is sex-linked recessive disorder, you know, due to defect in their in either the red or green cone of the eye, resulting in failure to discriminate between red and green color. So there are cone cells in our retina. Okay, so basically these cone cells get affected. So usually red cone is responsible for red light, you have blue cone, you have green cone responsible for understanding green and blue, right? Everything is there. But mostly people who have color blindness, they can have their either all the cone cells are affected, or probably red and green is affected, or red or blue is affected, or blue or green is affected. But the most common thing that you definitely see is what? Red green color blindness. Okay, what do you have, children? Ayo. Red and green color blindness. That is the most common thing. Fine. Now, yes. Now, this defect is due to mutation in certain genes present on the X chromosome. I told you already, this X chromosome is the one that is having problem. Now, it occurs in about 8% of males and only about 0.4% of females. You will get to know why is this the case. So, please remember this data that is given over here. First data is this, second data is this, that it occurs in 8% of the males, children. Okay. And what? 0.4% of the females. Okay, 8% of the males and 0.4% of the females. This is because the genes that lead to red-green color blindness are on the X chromosomes. And you know that males just have one X chromosome and females has two X chromosome. So in males, if this is defected, then obviously that person will be having, they do not have a choice. Whereas when you talk about females, if one is affected and the other one is not affected, this can be dominant over this. You know it's a recessive disease, right? So if there is a dominant allele over here, it can present itself and that person would not be affected. Which is why it is said that 8% of the males would have it, but 0.4%, just a little amount of females would be having this particular disease. It's a rare thing. Okay. Now, the son of a woman who carries the gene has 50% chance of being color blind. The son of a woman who carries the genes has 50% chance of being color blind. You will get to know why. The mother itself, mother is not herself color blind because the gene is recessive. That means that its effect is suppressed by the matching dominant. Oh, okay. Now listen carefully. Okay. Okay, I have given what is it affecting. So there is a retina over here in this particular region. You have what? The colored receptors, green, red and blue. Either of them can get affected, but mostly it is red and green that is getting affected. Now, suppose, okay, so there is XX from mother and just a second, children, I'll just shift a bit so that I can write everything here itself. So this is the first condition where there is XX and XY. Okay. This is the condition where the mother is the carrier. Okay. And the father is normal, children. The father is pretty normal. Now at this point of time, when I cross this with this, I get XCX. Okay. When I cross this with this, I get again X, CY, fine. Over this and this if I cross, I get X and X. This and this if I cross, I get X and Y. So in this, I am able to see that 25% chance is there for the female to be a carrier. Okay, 25% of the chance is there for the male to be deceased. That person can have the disease because there is no one to dominate, right? So this person, if it has one, then obviously he will be diseased. On the other hand, this will be normal. 25% of female will be what, children? Normal. And similarly, 25% of male will also be normal. Cool? Works. This is a case where the carrier mother is there and the normal father is there. Okay. Now, second case, where shall I write it? And plus, okay, I shall write it here. Okay. So over here, again the mother is carrier, I'll write C over here, okay, and the father is diseased, he is colorblind. At this point of time, what all chances are there that the progenies will have color blindness or not? So let's cross this and this. If we cross, I get XC, XC, okay. This and this if I cross, I get XC and Y. This and this if I cross, again I get XC and then X. This and this if I cross, I get X and Y. Fine. It's a female. Over here also you got the female, but the female was just a carrier because you had one X chromosome that was normal that could dominate over the other. Here you have both of them affected, so this 25% of the female will be diseased. 25% of the female will be children diseased, right? Over here, this is XC and there is Y. So this has to be again disease. So 25% of male is again children diseased. Fine. Now over here, there is a normal one to combat the effect of this. So 25% of the females will be children carrier. Okay, and this 25% of the male will be what children? Normal. Cool. This is how you have to keep on trying for different cases. They will be giving you a case where the mother is carrier and the father is this, or probably the mother's father was a carrier, then what is the case of this? You will get to see lots of questions like that. Now I will give one more scenario, children, since we are very close to all this thing. Let's study one more scenario where both the chromosomes of the mother is affected and over here the father is normal. Okay, both the chromosomes are affected and father is normal. So I say mother is colorblind whereas father is normal. Cool. Now let's cross. So come on. Okay. If this and this I cross, I get X, C, X, okay. If this and this I cross, I get X, C, Y, okay. If this and this I cross, I get X, C, X, okay. If this and this I cross, I get X, C, Y. In this case, when the mother is basically color blind, then obviously there is only one choice. This if it fuses with this, this if it fuses with this, anyway it will be this condition itself. Whatever you do, the condition will be this. So 50% of the times the male will be what children? The male will be deceased, okay, and 50% of the time, okay, the females will be just the carrier. Won't be disease but be the carrier. Now if you see, most of the times the males were the one that was who were having the disease. Every time the females who are there, even if they have it, mostly they are carriers. Mostly they are carriers, which is why it is said that over here the condition that is given is the son of a woman who carries the gene, okay, has 50% chance of being colorblind. This is the reason. So son of a mother who carry the genes will be having 50% of chance to be colorblind, diseased, right? Now one more thing is given, because the gene is recessive, yeah, that's why, because Y chromosome is there. Y chromosome has nothing to do with the normal production of genes, right? Now that means that its effect is suppressed by her matching dominant normal gene in the case of females. In the females, we have a matching dominant gene which covers everything up. A daughter will not normally be colorblind unless her mother is a carrier and the father is colorblind. So over here, the daughter will not be normally colorblind unless, okay, her mother is a carrier and the father is what? Colorblind. So here father is colorblind and the mother is a carrier, and you see 25% of the females, they are what? They are diseased. They are also colorblind. That's the only case. Rest of the time you saw that most of the times they are just carriers, children. Most of the times they are just carriers. I hope color blindness is paka clear to you. Okay. So please remember all these things, and also this. You have to practice a bit, but it will be really easy for you to understand if you keep on trying it. Okay. Now going ahead, what do we have next? Try this out. This is the second question that we have. Please tell me, ma'am, this is the second answer. Okay, if a colorblind man, the man is colorblind, man marries a woman who is homozygous for normal color vision, homozygous as in same, for what? Normal color vision. The probability of their son being colorblind is this. You can pause, okay? You can pause the video and write out by yourself. I will go ahead, but you can try it out by yourself. The answer you will get at the end of the lecture. Yeah. Now, talking about the next sex-linked recessive trait, which is known as hemophilia, and there are different types of hemophilia. One type of hemophilia is also known as Christmas disease. Why it is known as Christmas? It is too happy disease? No, children. The person who got it first, okay, where we could find that, okay, this mutation could also lead to what's a hemophilia. There was a person whose name, I forgot the first name, but the second name was Christmas. The last name was Christmas, which is why it was named as Christmas disease. So in your question paper, if it comes, please understand you're talking about hemophilia. You can remember hemophilia, the blood does not clot, blood is red in color. In Christmas also, everything is basically, everyone wears red color, the Santa Claus and everyone. Okay, you can think like that. Now, the sex-linked recessive disease, which shows the transmission from unaffected carrier female to some of the male progeny, has been widely studied. In this disease, a single protein that is a part of cascade of proteins involved in the clotting of blood is affected. So what do you mean by cascade of proteins? For example, A, B, C, D are the cascade of proteins which would be releasing what something that will lead to clotting of blood. But the B guy says that I am not going to produce what I was producing. So but when B is not doing its work, so team effect is not there. Obviously, this particular clotting factor which has to be produced is not being produced, which is why you have this particular disease. So a single gene, okay, single gene which is going to produce a protein, which is a cascade of proteins, which is a part of the cascade of proteins, will be getting affected in this over here. Okay, due to this, and in an affected individual, a simple cut will result in non-stop bleeding. That person will keep on bleeding because there is no clotting enzymes to stop it. Now, the heterozygous female, that is a carrier. Heterozygous, that means it is a carrier. Okay, heterozygous as in this is X, this is X. Here you have a gene for hemophilia, but here you have a normal. This is heterozygous. When you have similar, for example, this both genes are normal, you call them homozygous. Similarly, this both genes are affected, you call this homozygous, having both the genes as this thing, having both the defected genes. This is homozygous normal genes, this is homozygous defected genes that is getting over here. This is heterozygous, when something is different over here and the one is there to dominate it. Which is why, again, as I said, just like how there it was in the color blindness, this particular female who has a normal one will be dominating over this, and hence she will be just a carrier. She will be just a carrier, children. But on the other hand, the heterozygous female, as I said, will be a carrier and may transmit the disease to the son. Now for son, obviously, what do you need, children? I'm erasing, ayo, why do I always choose the wrong things? Just a second. I'm erasing this. This is X and this is Y chromosome of the father. Father has two chromosomes, X and Y, right? Now, I am saying that father is normal. Father does not have anything. The mother has the same thing. Now, you only think the gametes can contain either this X which does not have anything, so I am writing XN, that is normal, okay, or the gamete can be receiving XH, which is the one that has the mutation. Similarly, over here you get X, which is normal, and you get Y, which is again normal. Okay, suppose this Y can fuse with this, then it's all good, it's a normal child. Okay, normal male child. But if it fuses with this, what will happen, children? It's XH and Y, and that is normal. So this one, there is only one X. So whatever effect it has to show, it will be showing. So that particular son will be having that particular disease, whatever you do. What about this daughter's children? Again, if you fuse this with this, okay, you just have one way to produce a daughter. You fuse this with this, or you fuse this with this. If you fuse this, both of them, it will be normal daughter only, XN, XN, that is normal. But if you fuse this with this, what will happen? You get XN and you get XH. Again, there's a normal one to dominate over this, and this particular female will be a carrier, will be just a carrier will not show this effect of this particular disease. Being a carrier and showing the effect is totally different. So the males are always diseased in this condition. Okay, not always, basically you have 50% chance. If this fuses with this, then it could happen. If this fuses with this, then it won't be happening. Okay, so let's see, we'll make it and properly I'll show you. So the possibility of the female becoming a hemophilic is extremely rare, because the mother of such female has to be at least the carrier and the father should be hemophilic. Okay, the father also should be hemophilic. At this point of time, there are chances that this and this can come together and produce a diseased female, but that is kind of very, very rare. Okay, now because why? Because the father who is hemophilic, okay, the father, basically females, as I said, is a carrier mostly. Only males are the one who will be having this disease. Now we only think, if someone is having this disease, do you think that they can live for long if without clotting of their blood? No, they cannot live for long. So they would not be viable. So it's a rare case that you find a person who is still alive with hemophilia, and that person decides to mate with another person and might. So for example, unfortunately, that this woman is also a carrier and this guy is having that particular disease, that is kind of very, very rare, because this particular person might not live for long enough. Okay, because there is only one gene, there is no one to dominate over it and cover this problem up. No, there is no one to do that. Hence, this person will be having less viability, which is written right over here, unviable in the later stages of life. Not initially, but in the later stages of life. So they can go up to few years, but later on, there are high chances that things could change. They could pass away. Okay, the family pedigree of Q in Victoria shows a number of hemophilic descendants, as she was the carrier of the disease. If you know Q in Victoria, okay, she was a carrier of this particular disease. So we will make it and learn it. So you have X and H O U. Again, I will make it in a proper way only, so that my dear children remember it clearly. Just a second. Why is it not going? Okay. So over here you have X, XH, okay, the H is the affected one, and you have, it is, okay, instead of writing cross, I'll, okay, small cross, you have X and Y. So here the father is normal, okay, and the mother is basically a carrier. Mother is carrier, okay. You fuse this with this, you get XX. Okay, you fuse this with this, you get XY. You fuse this with this, you get XXH. Okay, you fuse this with this, you get XHY. Okay, so you have got two males. You have got two males, but only one male is affected. So I can say that 25% of the male, okay, out of 4, 25, 25, 25, 25 is equals to 100, right? So basically 25% of the males, they are diseased. 25% of the males, they are normal. Here also 25% of the females are normal, and 25% of the females are children carriers. Okay, they are what? Carriers. Now this is one scenario where the mother is the carrier and the father is normal. Now let's make the other scenario where, divide into 2 was, where it's X and the mother is hemophilic. Okay, so for mother to be hemophilic, you need both H, and then the father is normal. Assume father is normal. Okay, now at this point of time, I'll cross this and this. I get XHX. Okay, I should be not writing like this because this is not a dominant trait. Okay, just a second. Y is not working. Oh my god, I'm tired of this thing. Just give me a second, children. The pens, why they do not work at time and then when I need them. Okay, so over here when I fuse them, I get X and XH. Okay, and when I fuse this and this, I get XHY. Okay, when I fuse this with this, I get XXH again, and if I fuse this with this, I get XHY. So in this case, where the mother is hemophilic, and the father is again normal children, what do you find? Basically, here there is no one to dominate, here also there is no one to dominate. 25 plus 25, 50% of the times the males will be what? Diseased. 50% of the times the males will be diseased. Still the females, as you see, they are just carriers. 50% of these females are just carriers. Okay, this is the second scenario. Now if you want third scenario, let's make the third scenario where mother is also hemophilic, okay, and the father is also hemophilic, which is quite rare. Now in this case, whatever you do, if you fuse this with this, you get XH, XH, okay, and if you fuse this with this, you get XH, Y. If you fuse this with this, you get XH, XH again. Okay, if you fuse this with this, you get XH, and then you get Y. Okay, so if you see, this is disease, this is disease, this is also disease, this is also disease. All of them, may it be daughter, may it be sons, all of them, they are disease. 100% disease is being caused because of this, because there is no one to dominate in X over here. Okay, and here in XY there is only one X, and if it is affected, then you will have the disease. So basically, when the mother is hemophilic and the father is also hemophilic, the child has to be hemophilic anyways. So these are different combinations. Here you can try for yourself what will be the case where the mother is normal and the father is basically hemophilic. At this point of time, can you try by yourself and tell me the percentage, children? Can you try by yourself and tell me the percentage of the children who are going to be diseased? Okay, I am asking you, tell me the percentage of children who are going to be diseased. I am not asking about carriers. I am not asking about carriers. I am asking about the percentage, okay, of them who will be diseased. Okay, fine. Can you tell me the answer? So this is about hemophilia. Here the blood clotting enzymes basically are stopped, and as I have shown over here, the blood keeps on oozing out. Fine. So these are both sex-linked recessive diseases that you have to study. Now you have to try this out. This is the third question that I have for you. That is, a woman has X-linked condition on one of her X chromosome. This chromosome can be inherited by only daughters, only sons, only grandchildren, both sons and daughters. You have to try it out by yourself and do let me know the answer. This came in NEET 2018. At last we will be solving this. Okay. Now talking about sickle cell anemia. Now, sickle cell anemia, as I said, it is autosomal recessive disorder. It's not dominant. For dominant, we just have cystic fibrosis, okay, which is not there in your NCERT. Now, over here, as I said, first thing, autosome linked recessive trait that can be transmitted from parents to offspring when both the partners are carriers of the gene or heterozygous. Okay, when both has to be carriers of this gene. So let's see. First, so over here they have mentioned one more thing. It is maintained by a single pair of allele which is known as HBA, HBS. So over here just think that you have an allele, okay, on some chromosome, and this is the normal one which I am writing as HBA, okay, and this is not the normal one which I am writing as HBS. Okay, HBS. Now this is the case of mother. This is the mother's chromosome that she is sending down. Okay. Now on the other hand, I have fathers also. Father is again given the same thing where I have HBA, one normal gene, and there is one defected one, that is HBS. HBS stands for sickle cell anemia. S stands for sickle cell anemia. Okay. Now in gamete you can have either this or you can have this. Similarly you can have this or this. So I'll just directly fuse. Okay. If I fuse this with this, what I get, children? I get HBA and HBA again. Okay. This is normal. This is normal. There is no sickle cell anemia that is shown. Now if I fuse this with this, what do I get? I get HBA and then I get HBS. Fine. Does this person have sickle cell anemia? Do you think that they have sickle cell anemia? You tell me the answer, okay? Over here, I fuse with this and this, I get HBA and HBS. Fine. If I fuse this with this, I get HBS and I get HBS. At this point of time, tell me who all are diseased over here. Who all are diseased over here, children? Is there male, female and stuff like that over here? Can I say that this is this is a male, this is a female or anything like that? No. Come on. Okay. So, this is normal because there is only normal gene present over here. Okay. There is S, but there is A also. So, it will be combating over it. But still I can say that this, whoever gets this gene combination, they are the carriers. It can be male or female. Okay, it can be male or female. Just mothers are contributing this, fathers are contributing this. Okay, you have sex chromosome for sex determination. That is something different. It's about the other autosomes you're talking. Okay, you have got the autosomal copy where the one that is responsible for hemoglobin's chain formation, it is getting mutated in some or the other way. So, this person is the carrier. Won't be showing that disease of. Over here again I see that this particular person is the carrier. What about this? He or she, whoever it is, they are diseased. So out of 4 things, okay, I am talking about heterozygous condition where both over here you have different type of alleles. This is also different. This is not same is like this. Same is like this or this. This is not homozygous, this is heterozygous. So when father and mother, both of them are heterozygous, there are chance that 25% of the progenies will be diseased. Okay, 50% progenies will be children carrier. Okay, and again 25% will be what, children? Normal. It has nothing to do with the sex chromosome or the sex of the organism. That's why I'm telling you 10,000 times. Please remember it. It's an autosomal disease. Now, what happens in this autosomal disease? You need to know that as well. So, for that, I actually included something over here. Yeah, it's there. So, listen. What happens? We have our DNA, okay, and our DNA have codons, which we will be studying in next chapter. So, codons as in there is a group of things like A, U, G, there are few letters that comes together, okay. And now over here G, U, G, again you have G, U, G. So, listen, basically what happens, these codons are in the pair or in the form of 3 group. So, okay, I call this as a codon when they are 3 in number. And basically, they will be responsible for the formation of any amino acid. They will be responsible for formation of amino. Similarly, this will also form an amino acid. The other one will also form an amino acid. And when the amino acids, they come together, they form protein. They form a protein. So, in similarity to this, all the proteins are made in our body. If you talk about the structure of hemoglobin, we have 4 globin chains, children. We have 4 globin chains, 2 alpha and 2 beta. All of them are made up of proteins only. All of them are proteins only. So, the one that is responsible for formation of beta chain, you have 2 beta chain, and the one that is responsible for the formation of beta chain, the gene, okay, it has some mutation in it. It has some changes in the codons in it, such that initially there was GUG codon in the DNA, and this was coding for glutamic acid, which is an amino acid. Okay, so GUG codon was coding for glutamic acid. But now what happened, children? Basically, because of some mutation that has happened, the U over here, that is uracil over here, U stands for uracil, okay? It is now getting substituted by A, that is adenine. Okay, what is it, children? A, that is adenine. So, that is one big problem. Now, what will happen? The one thing that they were making initially will now be changed. It will now be changed. Okay, just a second. Just give me a second over here. I think I made a mistake. Substitution of glutamic acid by valine. Okay. Okay, that is the mistake that I did. My apologies, my apologies, my apologies. Just a second. Okay, that's what I was thinking, how it can be wrong. Okay, so initially it was GAG and this is GUG. My mistake. So over here, initially it was GAG only. Okay, but now what has happened? Come on. Okay. Now what has happened? This particular A, it is getting replaced by U, children. And why it is not happening? Okay, so it was A before, and now I am replacing it with U. Okay, now what will happen? This GAG, which was coding for glutamic acid, the glutamic acid won't be produced. Instead, GUG stands for valine. Valine will be produced. Now, valine is basically an amino acid, another amino acid. So, one amino acid change, that means the whole protein structure has changed. So, now this is a deformed form of beta chain. This is a deformed form of beta chain. Now, what will happen when the red blood cells, which is carrying the hemoglobin, faces a condition where there is low oxygen? Okay, faces the condition where there is low oxygen. At this point of time, usually our body is able to combat doing things. But over here, since the beta chain is also not normal, it is deformed, what will happen? The whole structure of the RBC becomes sickle shaped. Okay, have you seen a sickle? It looks like this. Okay, it becomes sickle shaped. Sickle shaped is used to sickle, like farmers use it. Okay, so it becomes in that particular shape, which is why it is known as sickle cell anemia. At this point of time, when your RBC is not in proper shape, do you think that it can do oxygen transport in a proper way? No. So these people who are diseased, that is 25% of the population, when this is the condition, okay, when they are diseased, they will be not able to survive. Okay, so now you might have doubt, ma'am, what if both of them, they are diseased? If they are diseased, they won't be able to survive for longer. Okay, if they are diseased, they won't be able to survive for longer with this particular condition in hand, which is why obviously the chances of them mating will be really less, because they won't be able to survive. So how can you expect them to mate? Okay, so that is one very, very important thing. And how you can remember this thing? How exactly did I remember that I was doing something wrong? Because children, it's not easy for anyone to remember a lot of things at times. But over here, see, A comes before G, right? So you can say GAG was the first thing that you had, and now A comes before U. That is what I meant. Okay, so you write GAG there and you write GUG here. Now again, G comes before V, so you write glutamic acid here. This is glutamic acid, and the second thing that you can write is valine. Okay, and this is on the 6th codon. Similarly, you will be having 5 more codons over here, and now on the 6th codon. This is 7th codon, next will be 8th codon. So on the 6th codon, this is happening, children. So you can remember G looks like 6, so it's on the 6th codon that this defect has happened, which is why now the beta chain is what deformed, children. It is deformed. Now let's read it once and then we'll go ahead. Okay. Okay, out of the 3 possible genotypes, only homozygous individuals for HBS show the disease phenotype, just how I showed you over here. Only them, they show the disease phenotype. Heterozygous HBS individuals appear apparently unaffected, but they are the carrier of the disease, as there is 50% possibility of transmission of the mutant gene to the progeny. So over here they are what? They are both carriers, and there is 50% of chance that they could give this particular thing to the next set of their progeny as well. Yeah, because again gametes will be found, one will be having this, one will be having this, so there is 50% chance. From this and this, 50% chance. From this and this, again 50% of chance for it to get things done. Okay. Now, just a second. Yes. The defect is caused by substitution of glutamic acid by valine at the 6th position of the beta globin chain of hemoglobin molecule. The substitution of amino acid in the globin protein results due to single base substitution. This is an example of single base substitution. So all these things that you see, you call them bases. And over here, you're not changing this and this. There's only a single base that is getting substituted with you. Okay, so this is an example of single base substitution as well. Now, at the 6th codon of the beta globin gene from GAG to GUG, the mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of RBC from biconcave disc to elongated sickle-like structure. So, usually it was like biconcave disc, as in it looked like this with a depth. Okay, it was like this, but now it has become like this. Okay, that is about sickle cell anemia. And over here it is a qualitative disorder. It is a very important information that I am giving you here. It is what, children? A qualitative disorder. It is not quantitative. Here the quantity of something is not getting disturbed, the quality of beta globin chain is getting disturbed. Okay, the quality of beta globin chain is affected. Fine, works. Now going ahead, what do we have? Yes, thalassemia, which is a quantitative disorder. This is quantitative because here the quantity of things would be affected. Let's read it once. Okay, so this is also an autosome linked recessive blood disease transmitted from parents to the offspring when both the partners are unaffected carrier for the gene. So again, if they are affected, they will also die. If both of them, they are affected, their disease, they will die. So obviously there is no chance of mating. But if they are carriers of this particular disease, then there are high chances that they can obviously provide it to the next generation that they are producing. Now the defect could be due to either mutation or deletion, which ultimately results in reduced rate of synthesis of the globin chains that make up the hemoglobin. So quantitative, as I said, it is reducing the quantity of both alpha chain or beta chain. So you have alpha thalassemia where you are affecting the alpha chain, you have beta thalassemia where you are affecting the beta chain of hemoglobin. Okay, it won't be making enough amount of beta chain or it won't be making enough amount of alpha chain. Now this causes the formation of abnormal hemoglobin molecules resulting into anemia. Obviously if you are not having enough amount of globin chain, how can you form the hemoglobin at first place, which is a characteristics of this disease. So anemia is caused, okay, where you do not have enough amount of iron, you do not have enough amount of hemoglobin. That is what anemia is all about, right? The hemoglobin structure needs all of this. If these things are not there, then obviously you get anemia. Thalassemia can be classified according to which chain of the hemoglobin molecules is affected. In alpha thalassemia, production of alpha globin chain is affected, while in beta thalassemia, production of beta globin chain is affected. Alpha thalassemia is controlled by two closely linked genes, HBA1 and HBA2 on chromosome 16 of each parent, and it is observed due to mutation or deletion of one or more of the 4 genes. So now listen, over here I'll explain here itself. Okay, so until now we were studying about this one gene getting affected everywhere. Okay, but over here imagine that this is the 16th chromosome. Okay, now on the 16th chromosome there will be 2 genes. So over here I have HBA1, this is also HBA1. Okay, I did a mistake. Okay, now this is HBA2, right, and this is HBA2 again. Now this particular disease is caused when either one of them is affected, one particular gene, or more of this particular genes are affected. If one is affected, then that means little amount of alpha hemoglobin will be not produced. Okay, if 2 genes are affected, that means little more amount of alpha hemoglobin chain won't be produced. If 4 of them are affected, then obviously there will be sharp decrease in the formation of alpha hemoglobin chain. It won't be produced at all. So that is what exactly is written over here. That is the closely linked genes, which is HBA1 and HBA2 on chromosome 16 of each parent. It's observed due to mutation or deletion of one or more of the 4 genes. These are the 4 genes, right? 1, 2, 3, 4. Even though this is allele, this is also allele, I call it 4 genes only. These are genes, no? 1, 2, 3, 4. Please remember 4, 4, 4, 4, very important thing. So the more genes affected, the less alpha hemoglobin molecules will be produced, while beta thalassemia is controlled by a single gene. So if you talk about beta thalassemia, this chromosome number 11, okay, and here there is only one gene that is going to get affected, that is HBB. HBB, you can remember hemoglobin beta, okay, and this is hemoglobin alpha. You can remember like that. Okay, now over here there's only one gene that is getting affected. If mutation is happening to this particular thing, obviously there will be less if mutation is happening to this also, then you can expect very less of what? This is for beta hemoglobin. Okay, I hope it's clear to you. So the parents, they can be carriers. If they are carriers, if they are carrying it, if there is having one mutation and one, this is a recessive one, so you just need one over here. If this is mutated, for example this is mutated, then still the other one is producing and they are known as the carriers. But suppose this and this, both of them are mutated, in this case obviously the disease will become me. The person, whoever who has it, will be having the disease. So if mutation is happening on one, then you're just a carrier, but if mutation is happening on both, then you are not just a carrier, you have the disease. Now, while beta thalassemia, as I said, is controlled by single gene on chromosome 11 of each parent and occurs due to mutation of one or both of the genes, thalassemia differs from sickle cell anemia in that the former is a quantitative problem of synthesizing too few hemoglobin molecules. As I said there, the beta globin chains are as a former, as in you're talking about this, okay. While the latter is a qualitative problem, you're talking about sickle cell anemia, that is messing up with the quality of the beta globin chain. Here you are messing up with the quantity, okay. Here you are messing up with the quantity of alpha and beta hemoglobin chain, which is why it is resulting in what? Anemia. There it was sickle cell shape, okay. So this is what you mean. So you have alpha and beta thalassemia. Again, that is minor and major. What do you call as minor? The ones that carries are known as minor. The one that have this disease, major. So things like that. Okay, I hope you got this. Now people can have it. It's a disease which you might or may not have. You might be a carrier, so you need to check it up. Okay, there is ways to check. So please check. So before getting married and all, nowadays people do a full body checkup regarding these genetic diseases as well, because obviously if a progeny has it, then they have to suffer a lot, right? So it is very obvious to have a normal checkup done. So anyone can do it at any age. If you think you are a carrier, probably if you think that in your family this something is having some problem is there, people are having certain issues, there are deaths being caused because of this thing, so you should be having that particular check in your mind, okay. So please do that. Now going ahead, okay, this is about beta thalassemia. I already explained it to you there itself, so I'm not explaining again. Yeah. Now this is the fourth question, I believe, right? Thalassemia and sickle cell anemia are caused due to the problem in globin chain molecule synthesis. Select the correct statement. Both are due to quantitative defect in globin chain. Thalassemia is due to less synthesis of globin molecules. Sickle cell anemia is due to a quantitative problem of globin molecules. Both are due to qualitative defect in globin chain synthesis. So please try it out for yourself. It will be really, really helpful. Okay. Now going ahead with phenylketonuria, which will be our last autosomal recessive disease. Okay. Apart from that, as I said, you have dystrophy. Okay, muscular dystrophy. That is an example of what? That is the example of same thing. Okay, recessive one, only autosomal recessive. So phenylketonuria, you already studied about this. Okay, you already studied about this. What happens basically? Usually on our DNA there is a gene which is responsible for the formation of phenylalanine hydroxylase. This is an enzyme. This is an enzyme, children. Phenylalanine hydroxylase, which is responsible for the conversion of phenylalanine, which is an amino acid, to what, children? To tyrosine. Okay, and this tyrosine will be used up for formation of melanin, a lot of things. Okay, it can be used for different purposes. But now this guy is not there. This guy is not there because there is some mutation in the genes that is responsible for formation of phenylalanine hydroxylase, that is an enzyme. So what will happen, children? This phenylalanine remains in our body and gets converted to something that is known as phenylpyruvic acid. It's get converted to something that is known as phenylpyruvic acid, which when stays in our body can lead to mental retardation. it will lead to what, children? Mental retardation. Okay, that is one particular character that you have to remember of this. Now this same thing we studied in pleiotropy, if you remember, which is why I said we already covered phenylketonuria. So in pleiotropy, what was happening? Since this particular enzyme is getting affected, apart from having this disease, so what is pleiotropy? Pleiotropy is one particular gene is responsible for multiple traits. So one particular gene, which is this, is responsible for multiple traits. So if it is affected, so multiple traits will also get affected. So here tyrosine is not being produced. Similarly, there will be less skin color, there will be less coloration in your hair, everything. The color changes happen. There is hypopigmentation. There is less amount of pigment in your skin and your hair. Okay, this is multiple traits. Your skin color is a trait, your hair color is a trait, and then this is also one thing. Okay, so you have 3 different traits being controlled by a single gene, because this single gene is contributing to this, which helps in the formation of this, and now this tyrosine is responsible for melanin, which is responsible for our pigmentation as well. Now keeping that away, as of now, let's not think of that, but just remember this is again a recessive disorder, autosomal linked recessive disorder that you get when you have, the disease is caused when both the parents are basically carriers. Both the parents have to be the carriers, then only can get disease, and there is only 25% of chance for having the disease. Okay, and this is the problem that is caused. And also one more thing, these are also excreted through the urine because of its poor absorption by kidney. So basically phenylpyruvic acid is also being excreted in the urine also because its absorption is not really great by our kidney. I hope it's clear. Okay. So that is about phenylketonuria, and with this we have covered all the Mendelian disorders which is given in your book. You tell me, wasn't it easy? Is it difficult for you to remember all these things, or was it easy? It's easy, right? Now since we know it, it's easy, but we have to solve the questions as well. Now talking about the next set of diseases, that is chromosomal diseases. I actually, just a second, okay? Oh, I think I deleted it somehow. Chromosomal diseases. Let me give you a brief about this, and then we'll go to Down syndrome, which is a part of this. Chromosomal disease, as I said, it is caused when there is either you have too much amount of one, if you have an extra chromosome, or you have one chromosome that is less. This is known as aneuploidy. Okay, this is known as aneuploidy. You can have less or you can have more. Now this is a chromosomal disorder. These are the chromosomal disorders that we are going to study. The first set of chromosomal disorder is what, children? Down syndrome. What exactly happens in Down syndrome? So basically we have, obviously for everything we have a pair of chromosome, right? So this is the 21st pair of chromosome. But over here in the case of Down syndrome, there will be one extra 21st chromosome over here. So instead of having just a pair, you are having one extra 21st chromosome, and since there are 3 chromosomes now, you call it trisomy. You call it what, children? Trisomy of, tri as in 3. Okay, it's known as trisomy of 21 chromosome. Now, what is the problem for this? What is the problem? Okay, so why it is known as Down syndrome? Because it was first described by Langdon Down in the year 1866. Okay, and the affected individual who was found, okay, he had a small stature, as in the height was really less, with small round face, furrowed tongue. So the tongue was like this, furrowed tongue, okay, and partially open mouth. So these are the features you need to know: small round face, furrowed tongue, and then partially open mouth. Palm is broad with characteristic palm crease, so there will be so many lines on the palm, and physical and psychomotor and mental development is retarded. This particular point is very much important, children. For Down syndrome, one basic feature that you can find is mental retardation. Okay, do not get confused with phenylketonuria. That is something different. That is a Mendelian disorder, a genetic disorder. Over here when talking about this is a chromosomal disease which is caused by trisomy of 21st chromosome. So among the 23 chromosomes that you have, there is one extra chromosome that gets added up over here. Probably during cell division something might have happened, which is why you have this. Okay, this is known as trisomy of 21st, and these are the things that you need to remember. And as I said, mental retardation is one of the most important point over here. Okay, going ahead. you have two other diseases, that is Klinefelter syndrome and Turner syndrome. So as you can see over here, this is a man and this is the female. This is a man and that is a female, but they have certain issues. Let's see what is the issue all about. So the genetic disease is also caused due to presence of an additional copy of X chromosome. So initially, just think that a person was having, so why does this happen? Basically, just imagine there is XX, okay, and there is XY children. Usually what should happen? You should have one X chromosome and one X chromosome in another gamete. Similarly, you should have one X chromosome over here, one X chromosome in another gamete. So these are the sperms and this should be the egg, right? But over here the situation is kind of different, where what is happening? Instead of giving one X chromosome towards that side, both the X chromosomes, okay, just a second, both the X chromosomes will be here itself. So what will happen now? What's the problem with that, ma'am? So when this egg fuses with this, you get XXX. Okay, this condition is not given in your textbook. It's for super feminism, so that is different. Let's just forget this. But this is given in your textbook where there is XXY. Okay, there should have been just an X and Y. If it was just X and Y, it would have been a male, but now you have XXY. Y, if it's there, it has to be a male, but anyhow, you have an extra X over here, which is why you say that this person has Klinefelter syndrome. So instead of having 46 chromosomes, that person has one extra chromosome. That means it makes a total of 47 chromosomes, my dear children. Okay, such an individual has overall masculine development since that person is a male, right? However, the feminine development of breast, that is gynecomastia, is also expressed. Such individuals are sterile. What do you mean by sterile? They cannot have babies. They are not fertile. They are sterile. So they will be having a masculine body, but then still they have some certain feminine characters such as development of breast, as you can see in this particular picture, and they will be also sterile. Now this development of breast you call it as gynecomastia. Okay, that is something different. And please remember, this is XXY with 47 number of chromosomes. Now when you talk about Turner syndrome, things are quite different there. What happens in Turner syndrome? Okay, so same thing, just imagine that this X chromosome, that is the sperm, fuses with this female gamete which does not have any chromosome, so you get this condition, that is XO. There is nothing. So actually you should be having 46 number of chromosome, but you have what, children? 45 number of chromosome, because you didn't get another chromosome from the gamete that is there in from the mother. Okay, so this become was 45. This is not 47, this is 45. I think I have written same thing for Turner syndrome also. My God. Okay, so basically this is the Klinefelter syndrome thingy that is present over here. So please don't mind that. Basically I will say what is Turner syndrome. Turner syndrome also, you will be having certain masculine characteristics, and again you will be also sterile. This particular female will also be sterile, children. Okay, so you know there is X chromosome, and X chromosome is responsible for the feminine character. So that person will be obviously a female, but with masculine character. Okay, and it will be XO, hence there will be just 45 number of chromosome, not more than that. Okay, I hope this is also clear to you. Fine. Now try this. This is the fifth question that you have. So these are the three what was I saying, chromosomal diseases or disorders that you need to study, which is there in your book. Okay, now Down syndrome in humans is due to what? You tell me. 3X chromosome, 3 copies of chromosome 21, monosomy. Monosomy is when you have one, okay, or 2Y chromosome, extra. So Since anyway we have to find the answer for all the questions, let me just come over here, but do let me know your answers if you have already solved there. So a woman has an X-linked, let's see who all got it correct. A woman has an X-linked condition on one of her X chromosome. This chromosome can be inherited by only daughters, only sons, only grandchildren, both sons and daughters. So the condition is the woman has X-linked condition on one of her chromosome. So there is some condition, I am writing XM on one of the chromosome, right? Now this chromosome can be inherited. They are not asking if the person will have the disease or not. They are telling about if this particular chromosome will be inherited by the daughter or the son. So mating it with a person, over here I have XX, okay. The next thing I have, so I will fuse this first instead of fusing this. So when I fuse this, I get XX, but this is mutated, and then I get this also. That means I get XY, that is mutated. So this is the female, this is a male. So I get both sons and daughter children. I get both the sons and daughter if there is an X-linked condition, because anyways X-linked as in X and Y makes a combination of XY, that is for males. So you have to pass it down both to female as well as the sons. Not every time, it's like 25% of the chance that the son will be having, 25% of the chance the female will also be having, rest 25-25 they will be normal, okay. Now next thing, if a color blind man marries a woman who is homozygous for normal color vision. So for a person to be color blind, I am writing the details over here, we will solve it in the next page, okay. For a person to be color blind, obviously this particular allele should be affected, this particular not allele, chromosome should be affected. So it is XC. Now the woman he is marrying is homozygous for normal color vision. So the woman does not have anything, she is just XX, okay. The probability of the son being color blind, you are talking about the son. Now let's solve it. Can we solve it? Shall I solve it here itself? Because if I add page, you won't be able to see it, but let me try. If we have one more page over here. Okay, I will solve it here. So the condition of the second question is like this, where there is X that is normal, and there is a female who has there is a male who is colorblind. So this is the male who is colorblind, this is the female who is not colorblind, and she is homozygous, as in same. She does not have any disease. Now, I get XXC, I get XY, I get XXC, and then I get XY. They were asking the son if he or he was colorblind or not. That was the question, right? How much percentage of, okay. What is the probability of their son being colorblind? You tell me. What is the probability, children, over here? If you see, what is the probability of the son being colorblind? Do they have this particular gene? Do they have this particular chromosome? So it's 0. The answer is what, children? 0. So who all got it 0? Who all got your correct answers? Children, my dear champions. Do you feel a boost of motivation when you solve these questions? I feel it when I make you solve these questions. And okay, I am doing something for you guys. Okay, next thing. Down syndrome in humans is due to 3 X chromosome, 3 copies of chromosome 21, monosomy to, this is a straight question, it's trisomy of chromosome number 21. So this is the answer. Now pick out the correct statement. Hemophilia is sex-linked recessive disease. Is it sex-linked recessive disease? Yes. It is a recessive disease, sex-linked also. Down syndrome is due to aneuploidy. There is addition or deletion of one chromosome. So yes, it is. Phenylketonuria is an autosomal recessive disease. Yes, it is autosomal and it is recessive. In autosomal recessive, we had sickle cell anemia, phenylketonuria and thalassemia, right? Sickle cell anemia is X-linked recessive gene disorder. Is it X-linked recessive gene disorder? No. Sickle cell anemia is autosomal recessive gene disorder. Okay, so basically this is correct. So 1, 2 and 3 is correct. NEET 2016 question, children. Okay, going ahead with the next question. Thalassemia and sickle cell anemia are caused due to a problem in the globin molecule synthesis. Agree? Yes. Select the correct statement. Both are due to quantitative defect in globin chain synthesis. No. One is due to quantitative defect, other is qualitative defect. Thalassemia is qualitative or quantitative? Thalassemia is quantitative, where there are 4 genes or probably 2 genes. If one of them or probably 4 of them gets decreased, the amount of hemoglobin chain is reduced. Whereas the other one, sickle cell anemia, is qualitative, where the beta globin chain is abnormal. It is messing up with the quality. So it is wrong. Thalassemia is due to less synthesis of globin molecules. Yes. Sickle cell anemia is due to quantitative problem. No. Now, both are due to qualitative defect. No. One is qualitative, so this is the correct answer. Okay. Thalassemia is due to less synthesis of globin molecules. Remember, HbA1, HbA2, you have two genes. Now, if both of them are getting affected or 4 of them getting affected, it will lessen the chance of alpha hemoglobin. Now, HBB genes are there, 2 of them. If one of them is getting affected, less chance of less formation of beta hemoglobin. Both of them are getting affected, then you know, right? So this is the condition. Now, one question for my dear little champions is the homework question that I have. A colorblind man marries a woman with normal sight who has no history of color blindness in her family. What is the probability of their grandson being colorblind? This is the homework question. If you have solved it, okay, successfully, do let me know. Like this, homework answer is this, ma'am. I'll be really happy to see that. Along with that, do let me know your comments. Do tell me, it's been long since I saw any of you talking to me probably about anything. No doubts, nothing is there. So I feel sad. So if you can, definitely do not hesitate. Ask me doubts. I'll be definitely happy to solve all your doubts. Yeah. And if you think that this particular video was helpful for you, then do not forget to subscribe to the channel, my dear children. God bless you all. Bye-bye. Take care.